Hello, this is Matt Lippert. I am the dairy agent in Clark and Wood counties in north central Wisconsin. And we're at Badger Dairy Insight today. And today our title is Corn Silage, Strong Suits and Complements. And pretty excited about talking about corn silage here in Wisconsin. We plant roughly 1 million acres, or I shouldn't plant, we harvest roughly 1 million acres of corn silage a year. And that makes us the #1 state in the country for corn silage. I know not everyone on board is necessarily from Wisconsin. Today is primary election day here in Wisconsin. And it's good today, but we're looking at quite a winter storm warning the next 2 days. Different areas of the state, quite a few areas getting over a foot of snow. So I'll introduce our first speaker for the day, which is Luiz Ferraretto. And he is going to talk about new insights about corn silage fatty acids. Luiz has done his research and extension interest in applied ruminant nutrition and management. And he's focused on understanding and improving starch and fiber utilization by dairy cows. And one of the focuses is corn silage, high moisture corn quality, and digestibility. So he's the man for this topic today. He comes to us with degrees from Sao Paulo University, PhD program here at UW-Madison, worked at the Minor Institute, was on staff at the University of Florida. And so I think with that, Luiz, you're well qualified to talk to us about this. So why don't you dive in about what we're having here, a fatty acid discussion in corn silage.
Yeah, thanks, Matt, for the introduction, and thank you all for attending our webinar series this afternoon. So this is a new topic related to corn silage for us. If you follow some of the UW extension work related to corn silage, you know that most of the time our goals would be to emphasize 2 of the main fractions associated with corn silage production. One of those will be the grain fraction, right, which is very rich in starch and help us to formulate diets based on energy density, depending on how you harvest, process, store, and formulate diets about that, right? In addition to that, we always discuss the stover fraction of corn silage, which is rich in fiber, or NDF, and also plays a major role on how to establish high forage diets, as well as intake of dry matter, passage rate, and consequently performance, right? However, I want to remind you today that in addition to those 2 main nutrients, starch and NDF, there are other nutrients in corn silage that, depending on what we do with our harvesting practice or storage practice, may change, right? So just to give you an example of that, here we have 2 independent data sets. One data set from Rock River Labs, another data set from Dairyland Labs. By the way, thank you both labs for sharing these 2 data sets with us. We really appreciate that. But if you pay attention to our red numbers here, you'll see that we highlighted total fatty acids. And yes, total fatty acids is a way smaller number than NDF or starch, but it's quite important. Said that, regardless of which analysis you use, which laboratory you use, how often you analyze total fatty acids, our goal today is to discuss the effects of common silage practice on long-chain fatty acids composition. Okay? So you may be wondering, but Luiz, why do you want to discuss that? Okay? Well, one of the key points that it's our job to discuss is how do we convert corn silage into meal components? And long-chain fatty acids may play a major role in that. Okay? And we'll discuss that in a second. So said that, let's try to understand a little bit more than total fatty acids concentration in corn silage. So this is a study conducted in collaboration between Penn State University and South Dakota State University. And basically, they use commercial hybrid trials, right? If you are familiar with university hybrid trials, very often seed companies, they entry a lot of their hybrids. So those hybrids are compared side by side. And then there are a lot of publications discussing yield, nutrient composition, nutrient digestibility, etc. So farmers can make more informed decision about which hybrids to use, right? And using those specific type of trials, those researchers, they decided to evaluate something that's quite unique in terms of corn silage data, which is fatty acid composition, right? So here you have a few of those fatty acids highlighted, and they are expressed as a total, as a percentage of total fatty acids, right? And I want to highlight to you one in particular, okay? The C18-2 or linoleic acid, okay? And the reason why I'm highlighting that is this will be key for our discussion today, because this is one of the fatty acids that, if not fully saturated throughout the biohydrogenation process in the rumen, it may end up to milk fat depression. But what I want to also mention about this specific study is that based on the proportion of C18-2 that they saw, they run a simple exercise to help us understand if you change corn silage percentage in the diet, okay? And if you use the average value that they found for C18-2 or the 10th or 90th percentile, how does that change, right? So for this specific example, they consider a dairy cow consuming about 55 pounds of dry matter per day. And you can see that if you add more corn silage to the diet, right, highlighted by the different colored bars, or if you have corn silage varying in the proportion of this specific fatty acid or the total concentration of this specific fatty acid, the cow will be consuming more of that, right? And what does that mean? It means that the more she consumes, more fatty acids have to be biohydrogenated in the rumen and very likely won't, right? It will end up very likely in the mammary gland, some of those intermediates, and possibly cause milk fat depression. So said that, let's try to understand a little bit more of this effect. And in order to do that, we will go back about 15 years or so, okay? So basically, in 2015, I had the opportunity to work with Dr. Shaver in a meta-analysis of hybrids used for corn silage production. And while searching the literature, we also saw that he had studies testing high oil corn hybrids, okay? Those were corn hybrids primarily used for ethanol production that were being tested for potential use as feed for dairy cows. And there are not a lot of trials with that. You soon will understand why. Okay, this is not very popular either. And I don't think there is any recent data testing for those, but this will help us to show you the potential effect of changing the amount of fatty acids in corn silage diets or corn silage based diets. So basically, when we did that, we separated all kernels or all hybrids with different kernels characteristics. And the two that we will discuss today are conventional, that sometimes were highlighted as yellow than corn, or those different types of high oil hybrids. If we pay attention to the nutrient composition of those hybrids, you see that they are fairly similar, right? Throughout those different studies that were summarized, dry matter was very similar because they were harvested obviously at very similar maturity, which is quite important because when you're comparing corn silage hybrids, they have to be harvested similarly so we can actually make inference about those hybrid effects. There is a small change in crude protein concentration. Fiber and starch are virtually the same, and either extract is slightly higher, right? Or you could say probably 50% higher for the high oil hybrids. So the question is, feeding a diet only different by the hybrid that you included there, conventional or high oil, how does the dairy cow will perform? And when we evaluate that response, there is no effect on intake or milk production, right? However, as we start going through milk components, you can see that fat and protein percentage were reduced when feeding those high oil hybrids, right? And if you pay attention to pounds per day of milk fat, those were reduced as well, right? And I think that's quite important because it shows that if you have high concentration of fat in the diet, there is potential you will be inducing milk fat depression, right? In this specific case, I'm unaware of studies showing difference in fatty acid profiles for those hybrids, but they are supposed to be the same, right? So said that is basically a quantity issue. But in addition to the composition of the corn silage, there are many other effects that may affect how the corn silage fatty acids may influence fatty acid production by the mammary gland, right? So if we shift gears a little bit, let's talk about hybrids that actually do not change fatty acids in the silage sample, but may change the potential for milk fat production, right? So if you use exactly the same study, okay, the same meta-analysis summarizing different types of hybrids now. Now we are going to discuss hybrids that vary in stock characteristics, right? Here are some of those hybrids. Brown midrib would be a key one, right? BMR are hybrids well known for lower lignin concentration. Then we have the conventional hybrids or dual purpose or isogenics that were used to compare to those other hybrids. High fiber digestibility hybrids are hybrids that we actually don't feed quite often, but we had some studies available, so we summarize as well. Those are hybrids that actually have a slightly higher NDF concentration, but the same lignin concentration. And because of that, those are more digestible, right? Because you change the lignin to NDF ratio. And then we have leafy hybrids. Those are hybrids that have more leaves above the ears, right? And theoretically, those leaves are more digestible than the stalk. This could also potentially be more digestible. If we compare the nutrient composition of those hybrids, we can see that dry matter, they are very similar. There are some minor effects on crude protein, but the key differences that we can see here would be lignin concentration, right? BMR hybrids have lower lignin than all the other hybrids. And then for fat, there are some minor effects between BMR and the high fiber digestibility being lower than the other two hybrids, okay? But those are very minor differences if we think about very normal variation in fat or either extract. And then obviously those hybrids with lower lignin, they have more digestible in vitro NDF, right? And to a certain extent, some of those hybrids have less starch. Okay, so if you think, or if you contrast with the previous example where we have corn silage with way more fat, now we have hybrids that are actually very similar or slightly lower here if we think about BMR and the high fiber digestibility, right? But let's see what the cows tell us about it. And if you are familiar with this data, I think it's very similar to what you may have heard about it. Hybrids that have greater fiber digestibility leads to greater consumption of dry matter, right? So here we can see cow's fat BMR or the high fiber digestibility hybrids, they eat about 2 pounds more per day, right? Compared with the other hybrids, and this will translate into greater milk production. However, pay attention to what happens with BMR, right? It has a reduction in percentage of milk fat, right? And this is a very typical response, right? However, you don't see a change in milk fat yield, right? You can see that in pounds per day is exactly the same production as the other hybrids. So if you allow me to speculate a little bit about that, and if you think about the intake, right? If you have exactly the same diet, milk, fat concentration in the silage is very similar. Basically, if the cow is consuming more, she is consuming more of those fatty acids, right? So very likely there is some sort of fact in the mammary gland inhibiting the production of milk fatty acids. And because of that, you see a small decrease in fat percentage, right? But then you are wondering, but Luiz, you have exactly the same amount of milk fat produced, right? In pounds per day. Yes, because you have to remember that milk fat is a combination of fatty acids produced by the mammary gland, fatty acids that were mobilized by body tissues or from body tissues, right? And fatty acids that are coming from the diet, right? So very likely some of this similar production is related to some of those extra fatty acids reaching the milk fat, right? But then you are wondering, but Luis, this is not depression, right? Because we produce the same. Very likely not, but there is some sort of inhibition in the mammary gland, right? Very likely there is less de novo fatty acids, which are the fatty acids produced by the mammary gland, 616 and lower, that probably are not there, right? So the reason why we presented that is not to criticize intake, but to show that there are multiple effects associated with diets that affect consumption of corn silage, consumption of those fatty acids, and the potential for milk fat depression, okay? So let's change to some other common practice that we know and sometimes implement on farm to change either nutrient composition or nutrient digestibility, but that could have some sort of effect on fatty acids in silage samples, right? So the first example here will be related to shot height, right, or cut height. So increasing cut height is a very well-known technique to change NDF. You basically reduce a little bit NDF and increase the starch. Basically, by increasing that, you are diluting more kernels into the same amount of kernels into less material of plant, right? And consequently, you dilute the NDF there, so you have a little bit more starch. But you also leave more lignin in the field, right? Because a lot of the lignin will be in the bottom part of the plant. So that's why you have greater fiber digestibility. So this could be a great tool to improve fiber digestibility. Unfortunately, this will be at the expense of yield, right? All the time you increase and leave more material in the field means you're harvesting less, right? But this is very well established. There are years that this is a very good tool to make sure you improve forage quality. Other years, forage inventory will not allow us to do that. But one question that we received a couple of years ago when discussing some of those effects, what does it do to fatty acids, right? And I'm unaware of other data sets evaluating that, but we ran a study testing fermentation and nutrient composition of cutting height, and then decide to reanalyze some of those samples for fatty acids, okay? And so basically what we saw is that as you increase cutting height, you basically will have a little bit more total fatty acids, but you're not gonna see a major change in fatty acid composition, right? So just to quickly explain how data will be presented here or is presented here, you will see that for each of those three fatty acids, C18-1, C18-2, C18-3, we have as a proportion of total fatty acids, and then between parentheses, we have as a percentage of dry matter, right? The p-values that we are presenting are related to the percentage of dry matter, right? So you can see that proportion minimally change from regular to the height shock, but the concentration in the dry matter changes because we are having a small increase in the total fatty acids, right? Well, why does that happen? So that study I mentioned from Pennsylvania and South Dakota, they actually separated different fractions of the plant, right? Leaves, husk, kernels, and the stalk. And what they saw is the vast majority of fatty acids, they actually come from kernels, right? So said that, we are diluting the same amount of kernels in less plant material. So that's very likely the reason for that. There's nothing physiological with the plant, is the simple fact that we are diluting more kernels into less plants, right? Or the same amount of kernels into less plants. And this has to be somehow accounted for, right? Another potential practice that changed the ratio between kernels and plant material is maturity harvest. Right, maturity harvest is actually a topic that we discuss since forever, right? I always joke that every 5 years we have to rerun silage trials with maturity to understand modern hybrids, but overall plant physiology remains the same, right? So we always have this question, should we keep plants longer in the field to harvest greater yields of dry matter and starch? And my answer to that is, it depends, right, on every dairy needs, but there are multiple things you have to pay attention to. One of those is you may increase the starch, and you may increase the amount of material you will be harvesting per acre, but this will come at the expense of slightly lower fiber digestibility, harder kernels that may not be properly processed, and lower starch digestibility because chemically the starch would be less available, right? But how about fatty acids? Today the goal is to discuss fatty acids. So in one of our studies, we collected samples from a single hybrid. Yes, I know it's a limited study and it only matters for this hybrid, but it's a good exercise to help us understand that. And we harvest this specific hybrid that was replicated into 4 different plots at 3 different maturities, a quarter, half, or 3/4 of kernel milk line. And before anybody asks me that, no, I don't suggest you to ever harvest corn at 1/4 of kernel milk line. We just harvest that maturity so we can have a good comparison and understand how it changes, right? So we call those early, intermediate, and late corn silage. Those were processed with either 1 or 3 millimeter rows. We're not going to discuss processing today, but those were also in silo for different time periods. So we had fresh samples or 0 day, and then silage samples in silo for 30, 120, or 240 days. And we will be discussing a storage length in a moment today. So the question that we tried to answer with this study, in addition to multiple other fermentation things, was, does it change fatty acids or amino acids? And today the focus is fatty acids, and I anticipate we didn't see much for amino acids. Okay? If we separate only the maturity fact, here we have total fatty acids, and as maturity progressed, we saw an increase in total fatty acids as percentage of dry matter. Okay? Here I'm presenting to you C18-0, C18-1, 2, and 3 as a percentage of fatty acids. And in this case, the proportion changed a little bit, but we didn't evaluate statistically. Okay, what we did evaluate statistically was as a percentage of dry matter, and because total fatty acids change, all of those change. Okay, so if we think about that, you can see that in addition to slightly increasing over time that you leave the material in the field, you also see a small increase in the proportion. Here it looks like you reduce a little bit C18-3 and increase a little bit C18-2 and C18-1, okay? And I think that's important because as you put into percentage of dry matter, there is a good amount of C18-2 that will be available in late maturity corn silage, okay? A question that we couldn't answer in this study, however, is if we wait until after 3/4, what happens? My best guess is that increase a little bit, but we don't have that information, okay? So when we first ran that study, based on these differences, we actually hypothesized that this was not a major effect, okay? And Ben Saylor, which was my PhD student during the time, during his defense, we had a nice exercise we went through. My colleague, João Doria, João is a faculty here at UW, and he did a lot of work with Lu Armentano on milk fat. And basically, João used one of the spreadsheets or one of the models that they developed together to see if he had any effect of maturity on potential milk fat depression. And what João did is he had the specific proportions of those fatty acids, unsaturated fatty acids, to the model and predict the amount of milk fatty acids. Okay? The flaw with this exercise is it was not corrected as we would in a diet, right? If we use late maturity corn silage, it has more starch, very likely you're gonna add less corn to the diet, right? That's not what was done here, but I think it helped us to explain what could possibly happen if this is not accounted for, right? So the blue line is the early maturity, the red line is the mid maturity, and the green line is the late maturity. The one to wait here is just a combination of processing and ensiling time, the storage from 0 to 240 days, okay? But you can see if you pay attention to maturity, there are more milk fatty acids when we feed the early maturity corn silage compared with the late maturity or the mid maturity, right? And if you pay attention to the fatty acids lower than C16, which are de novo fatty acids, those fatty acids that are actually synthesized by the mammary gland, they explain why, right? Again, this is not accounted for potential differences that we would have in diet formulation, but tell us that the fatty acid concentration in corn silage probably plays a major role if other parts of your management are not adequate, right? And then another important practice that we always work with related to corn silage is the potential improvements in starch digestibility associated with prolonged storage, right? This is a topic we have been discussing a lot. And we know that during ensiling, especially the initial 30 or 45 days, there's a spike in starch digestibility. Basically, bacteria fermenting silage secrete proteases that break down prolamine proteins surrounding starch, make starch more available, right? But this effect continues even after 30 or 45 days, but not to the same magnitude, right? And very often when we present this type of data, we receive the question, but how about the fatty acids? Is there any effect on fatty acids? And to date, we only run 2 studies about that, but one of these studies is this one that I just described to you with maturity and kernel processing, okay? And basically, Ben studies here, you can see that here we are presenting C18, 1, 2, and 3 as percentage of dry matter, okay? And you can see that from 0 to 30 days, there is a small increase in C18-2 and C18-3. Okay? However, there's nothing we can do about it, right? Everybody will have to ferment that silage. We cannot just feed fresh corn, right? But as it progresses from 30 to 120, 120 to 240, the effect is actually not as pronounced, right? You can see that there are some minor effects here from 30 to 120 days for C18-2, but in general, there's not much going on, right? So in order to figure out if this difference is relevant, we try to calculate. For a dairy cow consuming about 60 pounds of dry matter, right, per day, in a diet with about 50% corn silage, the difference between silage fermented for 30 days and 120 days would be about 9 grams of C18-2. Okay, I don't think this is relevant enough to cause any issues, okay, but I thought I would mention that as is a question we receive very often, and I think it should be considered. So implications of those results, right? There are many factors that contribute to milk fat depression. I barely discuss which factors are available, right? I mentioned intake. I mentioned fatty acid composition of feeds. In addition to that, there are forage NDF, NDF digestibility, starch concentration in the diet, starch digestibility, nutritional management of dairies, right? Avoiding slug feeding is a key factor there, right? And probably a lot of other factors that I may not even be thinking about, right? Genetic of dairy cows and several others, right? But if we think only about corn silage does contribute to the total unsaturated fatty acid load that the cow will consume. And if you think about it, most of our diets are actually corn silage based in the United States. Not all of them, but there is quite a good amount of corn silage being fed to dairy cows. Changes in fatty acid profile associated with corn silage practice, they are not major, okay? But they must be considered and corrected for the diet, especially if high levels of corn silage are being fed and if those changes in practice lead to greater intake. Okay? This is a very simple presentation about fatty acids. It's a very new topic. There's not a lot of information available about that, but it's something that we will continue to pursue and learn more about so we can make best informed decisions about the topic. Said that, I'd like to thank you again for joining this webinar this afternoon, and I'll be glad to answer any questions.
So Luis, you have some already. You can look on the chat, or I can read. I'll read the second one so far. When feeding high corn silage diets, over 25 pounds dry matter inclusion, should we measure fatty acids via wet chemistry? And then just the following comment, understanding that other fat sources in the diet are a huge part of the puzzle.
I think that if you have major changes in silage practice, would be wise analyzing for fatty acids, right? I didn't test NIR equations associated with fatty acids, but what I was told is that those NIR calibrations available commercially by different labs, they are actually reasonable, right? So you don't necessarily need to analyze via wet chemistry, right? You could have a simple NIR evaluation and have an idea of that, right? Because wet chemistry analysis, they are very expensive still for fatty acids. That's my understanding of that. And they take a little bit longer than NIR would, right? So that's what I would suggest, right? I also suggest making sure that we pay close attention to all the different corn sources we are feeding, right? Because I use corn silage as the example, but in addition to that, we also feed high moisture corn, corn grain, sometimes it's snaplage and corn byproducts, right? So I think there's a lot there that will contribute to that. I agree with you that understanding other fat sources are a huge part of that. So.
Okay, and just a reminder to folks, if you did have a question, you should type them in the chat and you'll find that logo on the bottom. So the other question came up right away as soon as you started talking, and it's about Plenish, which, as I understand, are high oleic acid soybeans. So any work on corn silage supplemented with that atypical type of soybean?
I'm unaware of any studies specifically related to either corn silage practice or levels of corn silage in the diet and Plenish soybeans. But my understanding of the initial research with those soybeans is that they actually help, right? Because you are reducing the amount of linoleic acid towards oleic acid and do really help with milk fat depression, right? I think a good question is, any chance we will have some sort of Plenish corn available, right? Or whatever other name people decide to give to that with high oleic corn that maybe help us to not having issues with very high levels of C18, 1, 2, and 3, or sorry, C18, 2, and 3 fat to dairy cows. I think that's the real question, right? And honestly, I have no idea if this will ever happen or not. I hope so.
So I have a question for you. You know, I guess I don't think we normally think about corn silage being an oil source, except for that we feed so much of it. And then you alluded to the fact that, well, we feed corn byproducts, we feed different types of corn, high moisture and corn, and then the corn silage. So what is the relative contribution in a typical diet from those different corn sources? Is just by the sheer amount of corn silage, is it one of the more major contributors of the oil from corn or not?
Corn silage will be one of the key contributors, right? I don't think it's the main contributors because we feed so many different things that have oil nowadays that I think it varies from diet to diet, right? But I do think it's important to pay attention first because we do feed a lot of corn silage, and also because corn silage is not one of the main fat sources that we consider, right? We never talk about corn silage as a fat source. So a lot of times we don't even know if there is variation or not, right? And I do think there is way more variation to silage samples than we know, right? I saw some, I presented some of the examples we know about, but I'm pretty sure there is a lot to learn there that will help us to figure that out, right? The other question that I think is important to understand is, how about when we have greater intakes, such as with BMR, right? Potentially with cutting height type of corn, right? What does it do to the total amount consumed? And very likely there is way more there, because you're not only increasing corn silage intake, but everything else, right? But this is driven by corn silage. So.
Okay, and we have, do you see fat variation in corn silage season to season or field to field?
Yeah, that's another great question. We didn't analyze field to field or season to season yet. We just start playing with different data sets to try to understand variations. So our initial assessment was to see what are the ranges or the normal range for those samples. And then we start running research with some of the key factors that we know that exist. Right. My best guess is that the same way that starch and MDF vary from field to field, you should expect changes in total fatty acid from field to field. I don't know if there is a difference in fatty acid profile. I think it's less likely than total fatty acids, right? One thing that is important to understand is that it looks like a lot of the variation is associated with having more starch, right? So basically having more kernels in a sample probably means having more fatty acids, because most of the fatty acids will be in the kernel fraction, right? So if you have a lot of kernel variation from field to field and season to season, very likely you're going to see those as well, right?
I know I'm familiar with a seed company that's marketing a high oil in the kernel, a higher oil corn. And I think the appeal is a lot to make perhaps organic producers that are trying to save on, trying to get more energy in their diet without purchasing outside sources. So I know there's genetic differences in corn and oil content. He's nodding yes. All right. Just one more question I had. I'm not sure if that was, I don't think it was fatty acid. You had that slide. It had a couple of your trials, and I think it was Kung in the corner. So was that a meta-analysis, I think? But you had that slide, and you had quite a difference in starch digestibility in several of your trials. And I guess we've been kind of led to believe, well, by the time you get out a year, everything kind of equilibrates. But it doesn't look to me like that's happening in the trials that you have done. That they don't get as high ever, it looks like to me, between the best case and the worst case.
You're absolutely right. And the initial studies we conducted with ensiling time or fermentation time, whatever you describe that, we actually hypothesized that at a certain point, samples will reach 100% of digestibility, right? So the less digestible corn would reach a similar level, right? And for most of the studies, this actually never happened, right? The most digestible material remained the most digestible, and the less digestible increase in digestibility but never reached the same extent, right? So I think this is a key factor there, and it's definitely true. We didn't see it reaching. I think what is important to understand is that fermentation is a great tool that help us to improve the digestibility of starch because it is more available, right? But here we'll be breaking down those prolamine proteins surrounding starch granules. But you also have to understand that less digestible material will remain less digestible, just going to increase a little bit, right? In terms of how long should we leave it there, it depends on year to year, depends on what is going on with your harvesting practice. I think the standard recommendation we have is about 3 to 4 months, if possible, of course, right? It's not easy to have more area to ensile 3 or 4 months carryover of silage, but if possible, that would be a good indication. And if you do that, make sure you have very good silage management, because the more you store, more likely to lose if you don't have good management.
Okay, thank you, Luis. I don't see any other questions for you.
Sounds good. Thank you.
Thank you very much.